Hi, everyone. Good afternoon. Let's get started. We will be doing lecture 9 today, which is the first lesson of Unit 3. Unit 3 being the last unit of the course. So we have the urinary system, and then next will be the musculoskeletal system. And then we will have the reproductive system and then the endocrine system, and that will wrap up the course. I'll tell you more about the quiz on next Tuesday when everybody is here. But let's begin by drawing our kidneys, okay? Just I mean, I know you guys had a busy week last week, right, with all the evaluations. So I'm not sure how much of you had a chance to watch the recordings. So let's just draw the kidney together and make sure that we are familiar with all the parts before we do the study guide questions. Okay, so please take out a piece of paper and we will create a kidneys together. All right. So first of all, we have the abdominal aorta here coming down, right? This is the abdominal aorta. Which is the one that kind of arches back from arches on top of the heart and then go backwards and down through your diaphragm, and then after passing through the diaphragm, the abdominal aorta is going to branch into the renal artery. And each of the renal artery will feed into your kidney. Okay, so that's one kidney here on the left side, and then you're going to have the other kidney on the right side. Okay, and you know, this blood vessel will continue to go down to the legs. And after the blood is filtered in the kidney, they will come back out and form the renal vein. Right, you will have the renal vein here as well coming out. And then it's going to go back up, right? It's going to be like kind of behind the aorta here, right? And it's going to go back to the heart through the inferior vena cava. Okay, so that's basically what it is. And each of the kidney is going to have a tube that comes out like that. And it's going to connect to your urinary bladder. Okay. Does anybody know what the name of these tubes are? Do you guys remember what those are called? They collect the urine and you know, guide them towards the urinary bladder here. Anybody? Yeah, exactly. They are the ureters. Rochelle, thank you. With an E though. Okay, so ureters. There are two ureters. Okay, one from each kidney, and it's going to go to the urinary bladder right here. Okay, and then it's going to stay there. The urine's going to stay in the urinary bladder for some time until it's ready to come out, and it's going to come out from the urethra. Okay, so everybody has two ureters, but one urethra, and then the pee comes out. All right, so in men, the urethra is longer compared to the female one. So that's you know, basically on a very gross level, right? What the urinary system looks like. Now we're going to zoom in into the kidneys and see more of the inside structures. So let's draw our nice kidney here. Let me try that one more time. It's not too nice, but it will have to do. Okay, maybe just try one more time. There we go. That's a little bit better. All right, so over here, the medial side of the kidney, there is a little indentation, a little depression, where all the blood vessels going to go in, come out, as well as the ureter is going to come out from there, okay? And this location we call the hilum. Okay, so again, it's where the blood vessel is going to go in, where the veins going to come out, and also you're going to have your ureter, right, coming out like that. So if you cut your kidney with a frontal section, you slice it into front and back portion, and you look at it, the inside will have all these little triangle or cone-shaped structures. Okay, so there's a cone-shaped structure right here. Just going to draw a few of them. Okay, there might be more than 3, but we'll just draw 3 to keep it simple. Okay. So each of these cone structures, they are called the renal. Renal means kidney, right? Renal pyramid. Okay, so you have a renal pyramid here, the renal pyramid here, and then an other renal pyramid here. In between the pyramids, you will have the renal column. Okay, so pyramid, column, pyramid, column, pyramid. And together, these things, which are found in the core of your kidney, on the inside of the kidney, that makes up your renal medulla. Okay, medulla means the inside. Okay. So the outside portion, like this portion here, this portion, that forms the outer part of your kidney, that would be the renal cortex. Okay, so this is the outer layer. This would be like the more like the core layer, the core, the inside of your kidney. All right. Now, one of the things you have to remember is in order for the kidney to work properly, the salt concentration of the medulla is progressively higher the deeper you go into the kidney. So for the renal cortex, it would be less salty. And the proper way to say it is it has lower osmolarity. Osmolarity. Oh, I forgot to turn on the transcript. Give me a second. There we go. Okay, so if you need to see the live caption, it's turned on now. So the outer part is going to be less salty, lower osmolarity compared to the renal medulla. That would have higher osmolarity. Okay. And I'll tell you later why this is important in terms of how the kidney functions. All right, what else do we have? At the tip here, at the tip of the pyramid, right, that little pointy part, that is called the renal papilla. A papilla is like a knob, right? Okay, like the pointy end. What happens is, outside of the papilla, you're gonna have a cavity, a space, right? The urine is actually gonna drip out from the papilla, kind of like, you know, when the ice when the ice is melting or something from the eavesdrop, you have all these icicles, and you know, on a warm day you can see the water kind of dripping as the icicle melts. That's kind of like that, okay? Except we're talking about urine here. The urine's going to drip out from the pointy end of the renal pyramid, and it's going to go into these cavities, okay, these empty spaces to collect them. So the immediate area outside of the renal papilla, the ones I'm coloring blue here, okay, those are called the minor calyx. Minor calyx. Okay. And then from the minor calyx, they are going to pool together and enter a bigger area, which I'm shading in with a darker blue here. That would be the major calyx. Major calyx right here. Okay. So as the urine's being produced, they dripped out from the renal papilla, they go into the minor calyx, and then they go into the major calyx. And then finally, it will go out into the renal pelvis before entering the ureter, okay? Renal pelvis, VIS. Okay, so it's going to go out and all the way to the urinary bladder. Does anybody have any questions so far about, anything that I've drawn? Okay, now there are a couple of other details here that we could add. The kidney actually is surrounded by a tough casing, okay, a fibrous casing. And that fibrous casing is called a renal capsule. And all this is in the recording, right? Okay, there is professional drawing that we labeled in the study guide at the towards the end of it, and the drawing is much nicer there, of course. Does anybody know what sits on top of the kidney? What organ sits on top of each of the kidney? I'm just going to draw it here with a different color, so it doesn't go all over this stuff. Anybody know what this is on top of the kidney?
Adrenal gland.
That's right. Okay, Gorinda, you got it right as well. This is the adrenal gland up here. Okay, so this whole thing is adrenal gland. You might have heard of adrenaline, right? So adrenaline is a hormone that comes out from the adrenal gland. Okay, there are other stuff that comes out from the adrenal gland as well, like aldosterone. If you finish the lecture recording, then we learned about the hormone aldosterone. So more on that later. But the adrenal gland sits on top of each of the kidney. All right. I hope that makes sense. So, in the kidney, we have something called the nephron. Okay, that's N-E-P-H-R-O-N, nephron. So I'm going to draw one nephron here. Okay, there's a ball at the front. It loops down into the medulla, come back out, and then like that. Okay, so this is not to scale. The nephrons are much smaller compared to the kidney. Okay, but this whole thing is a nephron. Okay, I'm going to draw it again here. Nephron. They are the functional unit of the kidney. In other words, the kidney is able to produce the urine, to filter your blood and produce the urine, because the nephrons are doing it on an individual basis. So you would have like millions of these nephrons in your kidney. Okay, so I'm going to draw another one here, for example. And they all go down a little bit and come back up, and then dip all the way to the papilla. So more on the nephron later on. So there you have it. That's an overview of the kidney structures. In case you didn't have a chance to watch the recordings yet. So I ask again, do you guys have any question before we move on? I think the usual students are here, right? Usually I get about like 15 to 16 students. And I think everyone is here. So I'm gonna tell you about the quiz. Quiz is gonna be next Tuesday. I'll post the information. And just to give you guys a chance to kind of boost your grade a little bit, I will let you have 2 attempts for the upcoming quiz. Okay, for quiz number what is it now? Quiz number 3. Okay, so for the quiz next Tuesday, I'm gonna give you 2 attempts, and the best score of the 2 attempts is going to count. Okay. Now in the past, people do the quiz and then immediately they do the second attempt. That's probably not the best strategy. Okay. I would recommend doing it in the morning and then study a little bit more if you didn't do too well, and then try it again. The second attempt is not mandatory. Okay, if you do it once and you like your mark, then you do not necessarily have to try the second time. Okay. So I hope this is gonna help a little bit. Oh, you're very welcome. All right, let's go back to the study guide here. All right, let's see what's going on here with this study guide situation. List 3 functions of the urinary system. So the urinary system is responsible for excretion. Okay, excretion. Yeah, the same availability, 6 to 11:59 or something like that. Yeah, everything is the same except you can do it twice. Okay, so excretion, which means remove wastes from blood. All right, so there's a lot of like waste product in the blood, right? A lot of stuff that we want to get rid of. Things like urea, there are like metabolic wastes, right? And all these things is not good if it builds up to a high level in the blood. So we want to get rid of it. But first we must extract them from the blood. So the process of removing these waste product from the blood and putting it elsewhere in the body, that's called excretion. It's important to remember the excretion does not take the waste out of the body. It just takes it out of the blood. It's still in the body. And then to actually get it out of the body, that's called elimination. Okay, so that would be to remove waste from body. Okay, so this excretion step happens at the nephron level, whereas the elimination, that's basically like the act of peeing, right? Urinating. That's physically removing waste from the body. So those are kind of the obvious functions for urinary system, but many people don't know that the kidney also plays an important role in homeostasis. Okay, so the kidney is important for water salt balance. Okay, if you haven't been drinking water, your kidney helps you conserve water. If you have too much salt level, it could cause your blood pressure to go up, and the kidney will play an active role in balancing the blood pressure as well. Okay, so we will specifically look at how it does these 2 things, as well as the ability to buffer the blood. Buffer means to resist change in pH. Your blood has to be around 7.35 in terms of pH. And if it gets too basic or too acidic, then it's very dangerous for the body. So your kidney plays a role in helping to maintain that narrow range of compatible pH. Okay, so there's a lot more things that it does in terms of homeostasis, but those are the 3 things that we will focus on in the lecture. Okay, so next we're gonna label the different parts of the nephron. I'm just gonna draw one over here on the side. You can draw it with me. So I know earlier when we were just sketching it, I did it like this, right? Okay, so now we're gonna do a slightly better version of it. Okay, so you can think of the nephron as a hollow tube, okay, almost like a straw. Okay, and it's usually very twisted up inside the body. Do you guys know what a crazy straw is? You know, like sometimes you see like fancy drinks or whatever, and then the straw comes in like all looped up like that. I don't know, something like that. Okay, so the nephron is kind of like that. It's really convoluted, very twisted up, and it packs itself in the nephron. But it's really difficult to label things when it's all twisted up like that, right? So we're gonna untwist it, and you just have to believe me that it will look something like this. Okay, so that is your not so crazy straw right here. Right, so there's always a ball thing at the front, and then it loops down, come back up, and then like this. Okay. So at the first part here, this ball is called the Bowman's capsule. Okay, which is right here. List the parts of the nephron in order that filtrate passes through. So the filtrate is talking about the stuff that's inside the nephron. Remember, it is a hollow structure. So inside it's filled with fluid, and the fluid is called the filtrate. For all intents and purposes, the filtrate is what will eventually become the urine. Okay, so filtrate eventually becomes urine. All right. So starting at the Bowman's capsule, we are going to go to the proximal tubule. Proximal tubule. Okay, which is this part right here, this immediate part. PT. Proximal tubule. Okay. So these filtrates will go here, and then it will go down. Make a U-turn and come right back up. So on the way down, this is called the descending limb. Descending limb. Followed by, of course, the opposite of descending is ascending. Ascending limb. Okay, so it makes a U-turn. And this loop here has a name. It is called the loop of Henle. Henle is the person who discovered it, presumably. Okay, so on the way down, on the way up together, it's the loop of Henle. Okay, so after coming up, it's going to go to this part right here. This is the distal tubule. The one that's closer to the ball, that's the proximal. The one that's further away, that is the distal tubule. And finally, it's going to go down this collecting duct. Collecting duct. Okay. Collecting ducts like that. Now, I don't know if you remember, but earlier when we were drawing this, I told you, right, there is a boundary, right? A boundary in the kidney, right? There's the outer portion and the inner portion. So the outer portion is called the renal cortex, whereas the inner portion is called the renal medulla, right? Okay, so the loop of Henle dips into the medulla and comes right back up before going in again. And so this collecting duct, like where does it go? You know, don't do this on your drawing. It's gonna mess it up, but I'm gonna do it because I can erase it easily. So look, right? Like let's say this is the renal pyramid, right? Remember this this little knob here? What is that called? Anybody remember what that's called? You can look back. I mean, assuming that you've copied it down earlier from me.
Renal papilla.
That's right, the
Papilla.
renal papilla. Yeah. So I forgot where I drew it. Right here. Okay, right. So you see this part here, right? Renal papilla. You see that the collecting duct goes all the way to the renal papilla, right? So that's what this is. This is the renal papilla. You know, it's like I imagine like eating watermelon, eh? Eating watermelon. There's the watermelon. Okay. And the sweetest part, it's the tip, right? Take a bite here. Oh, so sweet. That is like the renal papilla, the apex of it. Okay. So all these collecting duct is going to go all the way to the renal papilla, and then the filtrate, the yellow stuff, the urine, is going to come out from the renal papilla into the minor calyx and then the major calyx. Okay, so I hope the picture is coming together in terms of, you know, what's going on and how all these things fits together. So like I said, I'm just going to erase these. I don't want to make the note too cumbersome. Okay. So in reality, like I said, you would have millions and millions of these things. You're going to have, you know, Bowman's capsule, loop of Henle coming back up, go all the way down. You might have another one here, go down all the way. So all these nephrons will be busy filtering your blood, but ultimately all the filtrate, which contains all the waste product, will converge, will come together at the renal papilla. And then it will go out to, again, the minor calyx, so on and so forth. Are we good? All right, I hope so. Let's go back to this thing over here and see what else we can do. List all the structure a drop of urine would pass through after exiting the collecting duct. Okay, so I have been telling you, right, all day where to go. Let's just put it down. So from the collecting duct, it goes into, once again, the renal papilla. The renal papilla will drip the urine, the filtrate, into the minor calyx. Okay, sometimes you will see people write calyxes, that's the plural. This is singular. From minor, it goes to the major calyx. Then we will go into the renal pelvis. And remember the kidney, right? And then the thing comes out, right? That's the ureter. That's the renal pelvis. Is that big area immediately coming out from the kidney. And then it will go into the ureter, which takes it to the urinary bladder. Okay, don't just say bladder because you also have a gallbladder. We got to be specific. And after that, after the urinary bladder, it will go to the urethra before it comes out of the body. Okay. Next, we have to do the blood vessel. That is also a little bit of abstract concept. Let's draw it out together as well, okay, and try to understand it. So over here, we're going to draw a brand new fresh kidney. Brand new right here. I'm going to practice on drawing my kidney on the computer. There we go. Something like that. So I'm going to draw also a few of these pyramids. Okay. you should draw too. Okay, it doesn't have to be nice, but it should be big. If this is your paper, you know, your kidney should take up the whole page. All right, it's easier to label when it's big. All right, so work with me here, please. We have the big blood vessel coming down. Which one is this? Before branching into this one that goes into the kidney. So which one is this again? We labeled it earlier today. This one. That's right, Donna. That is the abdominal aorta. Very good. Okay, and then it will branch into the renal artery. Okay, one goes into the each kidney, right? One on each side. So I'll switch color every time they change name, all right? After the renal artery enters the hilum, H-I-L-U-M, the hilum of the kidney, they will branch into smaller ones. Branch into smaller ones. Okay, so these smaller ones, they are called the segmental artery. Little segments, okay? Segmental. Yeah, I'm just going to put A, all right? The segmental artery would approach the renal papilla, and when it kind of gets there, it's going to split into two smaller vessels that would go up the edges, the sides of your renal pyramid. Okay, so you would have another one that goes up the side like that. It goes up the side like that, right? So those vessels, they also have a name. They are called the interlobar artery. Interlobar. Then they will kind of join together on the outer edge, kind of at the boundary of the cortex and the medulla. Join together like that. Form an arch. That is called the arcuate, which means arch, artery. And if you're wondering, when is this going to end? Almost. Yeah, almost. So off the arcuate artery, you would have a bunch of smaller ones that comes up like that. All right, comes up like that. These ones are in the cortex, and they kind of, radiate out from the arcuate artery. So they are called the cortical radiate artery. Okay. I'm going to pause for a moment, just let you catch up, and in case you want to ask anything. Usually people would ask me in an actual, like in an in-person class, they would ask me, do we have to memorize this? Well, one of the advantages of online learning is you can have a reference sheet that lists the order, and you don't have to memorize as much, right? Okay, we're almost there. Now I'm gonna draw this thing again. Do you know what I'm drawing, guys? Can you tell me what this is? You should know what it is if you have been with the program.
Nephron.
That's right. That's a nephron. Thank you. Nephron, right? Yep. And we have millions of these within each kidney. All right. So again, you don't have to draw this, but I just want to remind you that a nephron would sit like right there, right? That's where the nephron would sit. Okay. And again, you have many of these. So I'm gonna pull one of these things out, one of these cortical radiate artery. I'm gonna pull one of them out just to make it bigger so we can see it clearly. From the cortical radiate artery, you actually have many more smaller ones that comes out from the side. Okay, and each one of these is gonna feed into the Bowman's capsule of the nephron. Okay, so these smaller ones that branches off from the cortical radiate artery, okay, these ones, they are called the afferent arteriole. Afferent arteriole. Okay, so the cortical radiate artery, the CRA, right? It's coming off from the cortex into the cortex, and on the CRA, you would have all these afferent arteriole. Each of those afferent arteriole will feed into the Bowman's capsule. Bowman's capsule of the nephron. All right, we are almost there. Just hang tight. Once it goes into the Bowman's capsule, the afferent arteriole will open up into a network of capillary. Okay, before exiting. So this network of capillary also has a name. It is called the glomerulus. There's a tongue twister right there. Glomerulus. Yeah, sure. Let me repeat that one more time. Okay. So each of these afferent arteriole will feed into the Bowman's capsule. Okay, so you will have one that goes into another Bowman's capsule. This will go into another Bowman's capsule. And as they enter the Bowman's capsule, the afferent arteriole will open up into a network of capillaries. The name of this capillary network is called the glomerulus. Glomerulus. Is that okay? Now I will explain this in more detail later on, but for now, just think back of what we learned in lecture 1, right? That seems like ages ago. What is one property that we learned about capillaries in general, right? If you have to describe the walls of the capillaries, what's the word that I kept on using back then? Hard to recall information from so far ago, right? The hippocampus not really working, eh? The word I kept on using back then was the fact that they are leaky. Remember, capillaries are leaky, right? That's why we have all these things oozing out of it, right? Okay, so regular capillaries are leaky, but the glomerulus are extra leaky. So when the blood goes through the glomerulus, you're gonna have all sorts of stuff being pushed out from the glomerulus. Okay, so these arrow, it represents things that are being pushed out. Now, when I say things, I don't necessarily means like blood, right? Okay, it's not the actual blood that get pushed out, but the components of the blood. Okay, the fluid component, which might contain waste product, it might even contain nutrients that we'll see. But the point is, as the blood is forced through the glomerulus, you have all these things coming out from it. And everything that comes out from the glomerulus will be captured by the Bowman's capsule, right? This ball here in the nephron, it will catch everything that comes out from the glomerulus. So all these things that comes out, what do they become? What do we call them now? Now that they're inside the nephron, what do we call them now? Do you remember the word that we use? Right? All these stuff that comes out, captured by the Bowman's capsule, which will continue on in the nephron. All these yellow stuff, we had a word for them. Do you remember what is it? Starts with an F. The filtrate, that's correct. So these are the filtrate. Okay, so at this point, the filtrate is not the final urine yet. There is actually some adjustment that we have to make. Okay, so in the different parts of the nephron, you know, the proximal tubule, the loop of Henle, all these different parts have different functions, and they will make adjustment to the filtrate before it comes out on the other end and become the urine. Okay, but essentially this is, you know, what's happening. You send the blood through these glomerulus, you squeeze out some of the contents of the blood, which then become the filtrate. But it's important to understand you do not squeeze out everything. Okay, otherwise it would be like peeing out blood, right? Which is of course not a good thing to have. So whatever we do not filter out here will leave the Bowman's capsule. And the vessel that's leaving is opposite to afferent arteriole. Well, you all learned it from me, right? Last unit. The opposite of afferent is what? I can hear you say it in your head. It is the efferent, right? Okay, so this is the efferent arteriole. All right. All right. Next, this part is a little bit weird, but it will open up into another web of capillary network. Okay, this one is really big. I'm just gonna draw it like that. It's really hard to draw it nicely, but there's another web. Okay, so this web is next to the nephron at all times. So we call it the peritubular. Okay, peri means next to the tube, in this case, the tube being the nephron, peritubular capillary. Okay. And we are there. We are done. Okay, watch. The peritubular capillary will actually go back like that. Okay, so next to each of these cortico-radiate artery is going to be a cortico-radiate vein. Okay, and these veins would be freshly connected to the peritubular capillaries. And then you would have a arcuate vein, right? Arcuate artery next to it will be the arcuate vein. And then you would have a blue one that comes back in. I'm not going to draw it because it's going to make the diagram very busy. It's going to come back in and you will have the interlobar vein. Okay? And for some reason, like nobody knows why, there is no segmental artery. It just skips directly to the renal vein and then up the inferior vena cava. So that's the blood flow and how it relates to the nephron. Okay. So this is the professional drawing. Okay, let's just watch this thing once together. You can just sit back and watch. I'll go back to the other slide in a bit. Okay, so we have the blood coming in, right, from the renal artery. It branches into these segmental artery, and then it goes up the side of the pyramid, right? Oh, this is not a good color. Maybe yellow. I don't know. Let's see. There we go. Goes up the side. That would be the when you think about it, it's the interlobar arteries. And then the outside, right? That's the cortico-radiate. Sorry, that's the arcuate artery. Where is that? Right here. Arcuate arteries, right? Arcuate arteries. Talking about the ones on the outside. And then coming off from these ones, right? You would have the cortico-radiate arteries. And then this diagram skips all the glomerular stuff that I told you. But the point is, it's going to go into the nephron, and then it's going to come back out afterwards. And when it does come back out, then you would have your cortico-radiate veins, right? So you see, for every artery, you're going to have a vein with the corresponding name. Okay, we have the interlobar vein somewhere right here. Okay, and then that will be next to the interlobar artery. Okay, it's just that for some reason, there is no segmental vein. Okay, I'm not sure why. It's just the way it is. There's the arcuate artery here, arcuate vein, renal vein, renal artery. Never mind the suprarenal artery. We don't need to know that. So that's basically it. Okay. And again, if you were with me earlier on, we can pull one of these CRA out, these cortical radii artery, and then you would have stuff that are coming off from it, right? And each of these things that are coming out will feed into the Bowman's capsule. Okay, so you see that thing that I just drew in hot pink? That's the afferent arteriole. It will go and become the glomerulus. Things will get filtered out of the glomerulus, and whatever came out will be captured by the Bowman's capsule. And then it will open up into an other web of blood vessels, right? I just drew it like that on the previous slide, right? Because it's hard to draw this. But this whole thing is called the peritubular. Let me zoom in here in case you cannot see it. The peritubular capillary before going back to the kidneys. So any question at all before we go back to the study guide? Seriously, like if you have anything, just ask. I hope that was somewhat manageable. Okay, thank you, Noor. All right, so let's write this down in a flow chart. All right, starting with the renal artery, we are gonna have segmental, right? Yeah, I know it's a lot to process, but the details, right? Like it's all written out here in a flow chart manner. So hopefully it will help you organize it a little bit better. But I do encourage you guys to actually draw it out yourself. I find that very helpful in terms of internalizing it and making it your own drawing to help you memorize it. Peritubular capillaries becomes the the peritubular capillaries becomes the cortical radii vein. Okay, so over here, let me show you again. All right, so the peritubular capillary will go over here. All right. So renal artery becomes the segmental artery. I'm just gonna use A for artery. Okay, so this is inside the kidney, right? And then they will go up the sides of the pyramid, forming the interlobar artery. And then from there, it will become the arcuate artery. Right. So going up the side, becoming the arcuate. And then we have all these crazy hair looking things. That would be the cortical radii artery. And then each of these crazy hair will have side branches. Those would be the afferent. Afferent means towards, right? Afferent arteriole this time. Smaller now. Which feeds into the Bowman's capsule. And inside the Bowman's capsule, it will open up into the network of capillaries, which we call the glomerulus. From the glomerulus, it will come out as the efferent arteriole. Before opening up again into the peritubular capillary. So if you think about it, it actually is like this. It's like artery, capillary, artery, capillary. It's two capillary network that are linked together in close proximity, right? One after the other. Okay, so after the peritubular capillary, we are on our way out. Right, we have the cortical radii vein. Okay, so CRA, and then now we have the CRV. And then we just copy it backwards, right? Arcuate, vein, and interlobar vein, and then we skip the segmental and just go straight to the renal vein. Okay. So I think there might be like question that ask you, you know, where does the blood go after the afferent arteriole? Then it's the glomerulus, right? So have this chart handy with you when you take the quiz, and they will help you, you know, answer some of the questions. Do I have any poll questions here? I do. They're so small. What's going on? Yeah, like this one, man. Let's try to do this one. Okay. Part of the skill that you have to learn is how to use information that are on your hands, right? To answer questions. It doesn't necessarily have to be from the memory per se. Okay. Yeah, I mean, most of you got it right. After the efferent arteriole, then it becomes the peritubular capillaries. Okay. It takes blood from the glomerulus, though. Okay, so you have to make sure to read the, I don't know what these things call it, to and the from, conjunction or something. You have to see what it is. To means where you're going next. From means where you were coming from before, right? Okay, so glomerulus goes into the efferent arteriole, and then it goes to the peritubular capillaries. Okay. All right. So that is that. So let's talk about the overall urine formation process. It basically has 3 steps in the urine formation process. We have the filtration part, the absorption, and the secretion. The analogy I use in class to explain this, it's like cleaning out a closet. Okay, I don't know how you clean out your closet, or if you do, but usually when I clean out my closet, I just take a bunch of stuff out from the closet and then I leave it on the floor. Okay, so that's kind of like the filtration process. You're gonna take a bunch of stuff from the blood, and then you're gonna put it into the filtrate. Right, you don't do much selection here. Okay, this is just a very a non-selective process. Right? A lot of things come out, and the things that you take out might actually end up being things that you want to keep. Okay, so imagine, right, like taking my whole pile of clothes and dumping it on the floor. Next thing I'm going to do is I'm going to go through my pile and I'm going to see what else I actually want to keep. Okay, like I don't want to throw away or donate everything on the floor, right? So I go through the clothing and then I pick the ones that I want to keep and then I put it back into the closet. Okay, so that's like absorption, right? In the nephron, you will examine the content of the filtrate and see what your body actually needs. What do you want to take back? It turns out there's a lot of stuff that we took out in the filtration step that we want to keep. Things like water, things like glucose, right? All these things we want to absorb as much as possible. Finally, as I'm putting some of the clothes back into my closet and folding it, hanging them, I might discover another piece of clothing that I no longer fit or no longer want. Then I will take that and then add it to the pile. That would be like the secretion process. Okay, along the way of the nephron, maybe there was some stuff that was in the blood that we missed in the filtration process. And now we want to add it to the waste. Now we want to add it to the urine. That happens in a secretion step. Okay, so to summarize, the filtration step happens first, and it's very non-selective, very crude, so to speak. Okay, there's no fine tuning to it. Absorption is to take back things that you want to keep in the body, and secretion is to get rid of things that you might have missed initially. All right. So the filtration process, where does it take place? What structure is involved? Okay, it's involved in the glomerulus. glomerulus plus Bowman's capsule. Okay. And in terms of, where things go, it's going from the blood to the filtrate. Okay, so we take the blood inside the glomerulus, we squeeze out some of the stuff, and they become the filtrate in the nephron. For absorption, this will happen in the proximal tubule. For the most part, it happens in the proximal tubule. 60 to 70% of the stuff that we lost in the filtration step will be reabsorbed in the proximal tubule. You will do a little bit of it at the loop of Henle, and then also in the distal tubule and collecting duct as well. Okay, so all these things inside these structures are the filtrate, and you will be absorbing them back to the peritubular capillary. Okay, so from the filtrate back to the blood. Okay, imagine having two side-by-side conveyor belt. One conveyor belt represent the nephron, the other represent the peritubular capillary. Because if you take a look at the picture here, right? Is it this one? Yeah. If you take a look at the picture, where's the pro one? Oh, there's the professional one. If you look at the peritubular capillary, is always right next to your nephron, right? So they're always side by side. And then you would have these transport protein that would take things from the filtrate and put it back into the blood. Okay, take things from the filtrate and put it back into the blood. These are things that we want to keep. Okay, we don't want to throw it away. But along the way, right, you might also see some things in the blood that doesn't belong there, and you want to get rid of it. So you also take that and then put it back in the filtrate. So along the entire length, right, of your nephron, you will always have some blood vessel next to it, and then there will be transport protein that kind of sort things out, right, reabsorbing it, right, secreting it, to make fine adjustment to your filtrate until it is ready to come out at the end. So secretion, that happens at the proximal tubule. No secretion in the loop of Henle, distal tubule and collecting duct, and it also involves the peritubular capillary. Okay, and the direction is going from blood back to the filtrate. Okay. Let's take a quick break, and when we come back, we will fill this in so you will have a better idea of exactly how urine is produced by each of these things. All right, so see you in about 5 minutes, guys. Hello again, let's keep going. So this table summarizes the events that are taking place inside each part of your nephron. So let's start with the renal corpuscle. And you're like, wait a minute, we have not talked about a renal corpuscle. And you will be correct. The renal corpuscle is actually a combination of two things. Okay, so it is the glomerulus plus the Bowman's capsule. Bowman is a name, right? This is a person, Bowman's capsule. Okay, so remember we have the afferent arteriole forming the glomerulus, the capillary network, and then exiting in the efferent arteriole. And this whole thing is surrounded by the Bowman's capsule. So the renal corpuscle is the Bowman's capsule plus the glomerulus. It's kind of like, bun plus sausage gives you hot dog. Okay, we just call the final thing something different. And renal corpuscle is a combination of these two things. So what happens in the renal corpuscle? What we have is called the glomerular filtration takes place. Okay, so as I tried to tell you earlier, the process is relatively non-selective. It is driven primarily by blood pressure. Okay. So normal blood pressure in capillaries, if you remember all the way from lecture 1, it's about 15 mmHg, right? Inside the glomerulus, it can go as high as 50 to 60 mmHg. Okay, so it's squeezing out all these blood through the leaky glomerulus, and as a result, things comes out. Right? So the walls of the glomerulus are leaky, right? Porous, which means leaky. Okay. So what comes out? Well, big things don't come out. Things like proteins and blood cells, like your white cells, red cells, white cell platelets, they remain within the blood. And then things like water, they come out. Electrolytes like sodium comes out. Glucose comes out. And maybe a little bit of like small amino acids. Okay, so Na plus, Cl minus. So these things, they're small enough to squeeze through the gaps in the capillary, and then they will come out. And when I say they come out, I mean they will come out of the glomerulus and be captured by the Bowman's capsule. Okay? And in the end, what do you get? The result of this process creates a protein-free plasma. You remember plasma? Right? Like if you take a blood and you spin it down in a centrifuge, you will have your hematocrit at the bottom, and then you will have the plasma at the top, right? So if you take the plasma and you minus all the protein, you take away all the protein, then you have a protein-free plasma, which is essentially what you would have got when things are filtered through your glomerulus. So all these things that are coming out, right? Protein-free plasma. All right. Now it's very important to remember, to understand, when we say water and sodium and chloride, they come out of the glomerulus, we do not mean all the water, right? Imagine if the blood goes in the afferent arteriole and all the water comes out, right? Then you will be left with like a sluggish chunk of like blood cells and proteins, right? And it wouldn't be able to move, right? Okay. So not everything comes out, but a high percentage of it, right? So some of the water comes out, and then whatever is not filtered will leave from the efferent arteriole. Do you understand what I mean by that? Okay, so go in, comes out, and the things that are leaking out include water, sodium chloride, glucose, and some, small amino acid perhaps. Okay, and this is not an exhaustive list, right? Okay, there are other electrolytes, right? Maybe potassium, calcium, whatever you have in your blood, they will come out over here. The key thing to remember is big things don't come out. What's considered big? Proteins, they're really big. Blood cells, they're really big. They all stay in the glomerulus, in the blood vessel. All right. So that's the first step. That's the filtration step. So let's take a look at what happens next. Next is the proximal tubule. And if you need a reminder, that's the Bowman's capsule, right? Loop of Henle and so on and so forth, right? This is the Bowman's capsule right here. This is the proximal tubule right here. The part right after your Bowman's capsule, okay? Just redraw that again, a little bit nicer. Okay, so we're at this part right here. So what's going on in the proximal tubule? Absorption. That's what's going to happen in the absorption. Right? You're going to take basically you wanna take back important stuff. Okay. All those water that we lost, we want to take them back. Glucose, we want to take them back. Okay. I probably should add here, right? And waste product. I don't want to make it sound like a waste. I don't want to make it sound like that, only the good stuff leaks out. There are some waste product that leaked out as well. But the point is, along with the waste, we actually lost a lot of important useful stuff as well. So it's time to take it back. Okay. So approximately 60 to 70% of the filtrate will be returned to the blood at this point. Okay, so everything that came out in the first step, we're going to take back 60 to 70% of it. This process involves both passive and active transport, and it is tightly regulated by hormones. Okay. Now, let me before we fill in this next part here, let me try to explain something to you. So I'm gonna zoom in the proximal tubule right here. Okay, and inside we have the filtrate, the yellow stuff over here. That's the filtrate. And dissolved in the filtrate, there are like many different things, right? Like I said, there could be electrolytes, there could be glucose. So I'm gonna put some green dots here to represent glucose. Okay. Next to the proximal tubule, we will have a blood vessel, right? That would be like the peritubular capillary. Maybe I'll just write out peritubular capillary. Okay. Now just watch first, okay? See if you know what I'm doing. In order for these glucose to go back into the blood, you're gonna need a glucose transporter. Okay? A membrane protein that will grab the glucose and then put it back into the blood. Okay. Remember that picture I was trying to tell you about, like this not picture, but like analogy, right? Like two conveyor belt, right? We have the proximal tubule on one end, we have the peritubular capillaries at the other end. And between the two conveyor belt would be like, a bunch of workers like myself. And my job is to check the stuff that are passing by. And every time I see like a glucose, I would have to grab the glucose and put it back into the blood. Okay, so try to visualize that. Okay, so you have all these workers down the line, and then they are all trying to look for glucose and grab onto it and put it back to the blood. So the number of workers, the number of carrier proteins is not infinite. Okay, you only have a fixed amount of these carrier proteins. And if all of them are already working at maximum capacity, if they're all working as fast as they can, and you have too many glucose going by, then what happens, right? You will inevitably lose some of the glucose, right? You won't be able to grab all the glucose and returning them back into the blood. Do you understand? So how much you can take back, the amount of substances that can be taken back depends on the number of carriers available. If all the carriers are like working at maximum speed, maximum capacity, you won't be able to absorb beyond a certain point. Okay, and that is what we call the threshold level. Now, the threshold level will be different for different substances. Some substances you will have more carrier available for it. Some you will not have as much. So the threshold level is different. But the point is, there's going to be some kind of maximum amount that you can take back. All right. So let me try to apply this for you. All right. So normally this is not going to be a problem, but for people, what kind of condition causes someone to have a lot of sugar in their blood? Anybody?
Diabetes.
Yeah, exactly. Right. So people who are diabetic, either because of a low insulin level or insensitivity to insulin, they will have a lot of glucose in their blood. So what happens is in the filtration step, all these glucose would come out of the glomerulus and they would end up in the filtrate. Okay. And you were supposed to take these glucose back, right? Into the blood, but if all the glucose transporter are already working at maximum capacity, then there will be some glucose that are left in the filtrate. Okay. And eventually the filtrate becomes the urine, and that's why diabetic patients, if left untreated, they would have sugary urine, right? In fact, the doctor could take one of those strips and dip it into the urine to test for the presence of glucose. Do you understand that? Okay. So 2 takeaway points here. Number 1, we want to take back as much as we can, but in order to absorb it, we must have some kind of carrier. And number 2, you can only take things back if you have a carrier. For some things like water and sodium chloride, they can rely on passive transport. But if you are drinking coffee, for example, caffeine, right? There is no natural carrier for caffeine, because that's not something that your body expects to be in your filtrate, right? So all the caffeine will stay within the filtrate and it will not go back to the blood, right? So for those of you who drink coffee, right, you will know after you drink coffee and you go pee, your pee actually smells a bit like coffee, right? The caffeine stuff. That's because all that caffeine comes out of your urine, right? They never got like reabsorbed. All right. Any question before I move on to the last point? Okay. Blood pH is controlled by secretion of hydrogen ion and absorption of bicarbonate. All right. So I'm just gonna quickly show you this chemical equation and see if you understand it. We produce carbon dioxide, as you know, because of cellular respiration, and that will combine with the water that is in our body, in the bloodstream. And what's the end product? Does anybody know? Did you guys learn this in chemistry? Carbon dioxide plus water, what does that give you? No? Who's your chemistry teacher? Anyways, I forgot to ask you guys. What is the product here? CO2 plus H2O. That's okay. It is H2CO3, carbonic acid. Hopefully that rings a bell. And then the H2CO3 is actually going to be broken down into H plus and HCO3. Okay. Now, what they sometimes don't tell you in chemistry class is chemical reaction can go both ways. Okay? It doesn't always have to go one way. It could go, both ways. And the general thing to remember is more H plus equals to lower pH. And less H plus equals to more higher pH. Okay, lower pH is acidic, huh? And then higher pH is basic. Okay. So the blood pH, as I said, needs to be controlled. So how is this helping? Well, if we have too much H plus in our blood, too much H plus, that means it is too acidic, right? So what do? We can do secretion of H plus. We can take the H plus from the blood and then we can get rid of it. Okay, so by removing H plus from the blood, you will make the blood less acidic. Another thing we could do is we can absorb bicarbonate. We can take back bicarbonate. So the bicarbonate, the H3CO3, the extra one that we absorb, will combine with the H plus, kind of mop them up and turn it back into H2CO3. So by actively getting rid of H plus and then taking HCO3 and combining it with H plus, we can lower the acidity of the blood. Okay, so that's how the kidney helps buffer your blood. Does that make sense? Okay. Let me know if you have questions, okay? Just ask anytime. Then after the proximal tubule, we're going to go down the descending limb of the loop of Henle. All right. Now, you really going to have to watch the video for the full explanation here, okay? I'm just going to have to briefly explain it here for you. Okay, so the loop of Henle has two parts, right? There is the descending limb and then there is the ascending limb. All right, so here's the filtrate that's coming off from the proximal tubule. We already absorbed like a bunch of stuff, okay? 60-70% of it was absorbed, but there's still a lot of things we can take back. And so what happens is on the way down, we are going to absorb water. Okay, let me try to use a different color here. Okay, to help. Oh my goodness, I just erased the whole thing. I'm just gonna use a different color here to help you see what I mean. Okay, so on the way down, we will be absorbing water. Okay, so you start with filtrate that are not too concentrated, but as you go down, as you absorb more and more water, the filtrate gets more and more concentrated. Okay, as I'm trying to show you with darker and darker color. Do you understand? Okay, so as filtrate goes down, water is being absorbed and it gets more and more concentrated. Right? So the descending limb of the loop of Henle, this section of the nephron is impermeable to sodium and chloride. Okay. As filtrate moves along the descending limb, water is absorbed through osmosis. Okay, because I was telling you like earlier today, as you go deeper and deeper into the medulla, the surrounding area gets saltier and saltier, right? So all those salty environment will help suck the water out of your nephron. The concentration of filtrate increases. Okay, so you're taking water out as you go down. Right, so it is the most concentrated at the bottom of the loop. Now on the way up, we are going to be absorbing sodium chloride. Okay, so as you remove sodium chloride from it, it will now become less salty, right? The water is not moving anymore on the way up. You're just taking back the salt. Okay, so it will get lighter in terms of the concentration. Lighter and lighter, like that. Okay. So you absorb water on the way down, you absorb sodium chloride on the way up. Okay, so in terms of like osmolarity, I can give you some numbers to see. Let's say this is 400 unit. On the way down, it will become like 600, maybe 800, and then like, 1200 or something. But on the way back up, because we're taking out the salt, then it will lower once again. And then you might go back to 400. Okay. So ascending limb of the loop of Henle. As filtrate moves along the ascending limb, sodium and chloride is absorbed by active transport. The concentration of the filtrate decreases as it goes up. So that takes us to the last two parts, the distal tubule and the collecting duct. Okay, so these locations are impermeable to solids, and adjustment can only be made by hormones. the amount of substances absorbed or secreted is controlled by various hormones, such as aldosterone, anti-diuretic hormone, and it's missing a blank here, and atrial natriuretic hormone. Okay, we'll talk about these in a bit if you haven't watched the video yet. Okay, so that's it. That's how you make urine. It's happening continuously, 24/7. All right, let's take a look at these question right here. Write the chemical equation for bicarbonate buffer system found in blood. So I sort of explained that earlier. It is the H2CO3. Okay, we put AQ because it's dissolved in our blood. It's aqueous. And the reaction can go forward or backwards. So the H2CO3 will break down into H+ as well as bicarbonate. You might remember bicarbonate from the digestive system, right? Bicarbonate is, the sodium bicarbonate. It neutralizes the acid. Okay, so just to reiterate, buffer resist small changes in pH. Okay, if you change it by too much, then you will destroy the buffer system, and it won't work anymore. And the blood pH is approximately 7.35. You cannot go lower than 7.3. You cannot go higher than 7.4. It is very dangerous. You might slip into a coma. Okay. Explain how the buffer system resists changes in pH. So there are two scenarios. First, when pH increases. Okay, so when the pH is basic, in other words, that means you have not enough H+. Okay, and what happens if you don't have enough H+? The buffer reaction shifts. Which direction does it go? If you don't have enough H+, do you want to move towards the right or you want to move towards the left? If we do not have enough H+. Yeah, exactly, Mavis. The buffer reaction shifts towards the right side to produce more H+. Right, Ahmed? If you go shift to the right, you make more of what you need, right? If you shift towards the left, then you end up using the H+. Okay, we don't want to use them. We don't have enough. Okay. On the other hand, when pH decreases, it's too acidic. Okay, too much H+, the buffer reaction shifts towards the left to use up or to remove excess H+. Now just pause for a second and listen to what I have to say. This happens in the blood. It has nothing, absolutely nothing to do with the kidney. However, sometimes the blood buffer is not enough. Okay, maybe for some reason, okay, back to the diabetic situation. For someone who's diabetic, they actually would turn to fat metabolism. Okay, and that generates a lot of ketones in the body. You might have learned about ketones in chemistry, right? So when you have a lot of ketones, right, it could lead to ketosis. And one of the side effects of ketosis is your blood pH is going to drop. So in that situation, the buffer system in the blood is not able to cope with the drastic change. And that's when the kidney needs to come in to help, right? So rather than just relying on the blood to mop up the H plus on its own, the kidney will help remove the excess H plus by, pumping it away at the proximal tubule, or putting it in the urine, right? So it makes your pee more acidic, which is fine because you're going to pee it out anyways, right? Okay. And you will take back the HCO3 to help mop up additional H plus. So explain how the kidney plays a role in maintaining the buffer system at the proximal tubule. Excess H plus can be secreted. And additional HCO3 can be absorbed, right? So extra HCO3 will combine with excess H plus to shift the reaction towards the left. Okay, by shifting it to the left, we will help decrease the excess H plus. Okay. Are you guys still with me? How's your attention span? Are you able to learn the next big thing? The next thing is also very confusing. But fortunately, there is a recording for it. That one is a little bit dated. I did the recording like several years ago. But it's still the same stuff. It hasn't changed much since then. Okay, so if you're all caught up, we're gonna move on. Use the flowchart to illustrate how the RAS, R-A-A-S. Do you have a question, Anike? Or you just press unmute accidentally? Oh, okay. How the RAS, explain how the role of RAS, I think this is grammatically wrong. Use the flowchart to illustrate how RAS, we'll say how RAS maintains blood pressure. Okay, maintains blood pressure. So what the heck is RAS, right? So RAS stands for renin angiotensin aldosterone system. Okay, RAS, R-A-A-S. What is the purpose? The purpose of RAS is to increase blood pressure. Okay, please remember that. Purpose of RAS is to increase blood pressure. All right. So normally the RAS is the system, right? Is not activated. Okay, we need to trigger it. And the trigger for RAS is low blood pressure, drop in blood pressure. Can someone give me some examples of why we have low blood pressure? Like what are some like situations that could lead to low blood pressure? Doesn't have to be like long-term chronic diseases, like just like normal things that we experience every day that could lead to drop in blood pressure. Yeah, dehydration, right? Dehydration. Like my kids, I don't get them. I pack them a full water bottle and then they just bring the whole water bottle back. And I'm like, don't you guys drink? It's like, it's so hot outside. I don't know how they manage to not get dehydrated. And Mavis said blood loss. That's another one as well. Very good. Blood loss. And we talk about like excessive blood loss, right? Like in if you suffer like a very serious injury, like a car accident, for example, and you're bleeding out, that could lead to drop in blood pressure. Okay, so a drop in blood pressure is gonna trigger the RAS. Okay, because the situation requires you to elevate your blood pressure. So where does it all start? Well, hopefully you know what I'm drawing now, because I've been drawing this for the whole class. That here is the glomerulus, right? And the glomerulus is surrounded by this guy, which is the Bowman's capsule. You probably know by now that this is the afferent arteriole. Nothing new so far. Okay. What's new is this next thing here. Okay. You actually have some special cells over here. And for simplicity, I'm just gonna call it the JGA. Okay. Juxtaglomerular apparatus. It's in your notes. Okay. It's apparatus means a group of cells that are located next to the glomerulus. Juxta is next to. Juxtaglomerular apparatus, or JGA. All right. So here's what's going on. You experience a drop in blood pressure. That will cause the blood flow to the glomerulus to be reduced. And the JGA is going to detect that sudden drop in blood flow. And in response to the drop in blood pressure, the JGA is going to release an enzyme called renin. So that's the R in the RAS, the renin. Okay, it is an enzyme. Okay, no renin unless you have drop in blood pressure. Okay. All right, so we'll leave that alone for now. Let's move on to the liver. There's your liver, and you know where the liver is now. We learned that in the digestive system. The liver produces a lot of plasma proteins, right? We learned that from lecture 2. There's albumin, fibrinogen, so on and so forth. And in the context of what we're talking about, there is something called angiotensinogen. Very long name. Angio like the heart. 10, number 10. Sin, when you do something bad. Ogen. Okay, ogen means it's like inactive, right? So the angiotensinogen is inactive. Right? Do you have angiotensinogen in your blood right now? Probably. Okay, but that's okay. They're not doing anything because they're inactive. All right. So when do we want to turn them on? Well, we're going to turn them on when you have low blood pressure. When you have low blood pressure, the renin will come out from the JGA. And you probably guessed it, the renin will take the angiotensinogen and cut it. And it will become angiotensin number 1. All right. Number 1 is not the end, unfortunately. You will have to activate it one more time, and it will become angiotensin number 2. But in order for us to do that, we will need another enzyme. This time, the enzyme will be coming from, hopefully you can tell what I'm drawing, the lungs. So the lungs will produce an enzyme called ACE, which stands for angiotensin converting enzyme. Okay, I'm going to give you a moment to take this all in, and please ask questions if you have any. Okay. So what's the problem here? Right? The problem is we are dehydrated. We might be losing blood, and our blood pressure is dropping. So ultimately, all these things has to result in an increase in blood pressure, right? So the RAS system, right? We have the R now. That's the renin. We got the angiotensin going. And the last A here, do you remember what the last A represent? Did you copy that down? What is that?
Aldosterone.
That is the aldosterone. That's correct. So the angiotensin will actually do two things. Okay, we'll do the easier one first. The angiotensin will cause vasoconstriction. Right, it will cause your blood vessels to squeeze down, to become smaller. Right, we learned in lecture 1, when your blood vessel gets smaller, your blood pressure goes up, right? So vasoconstriction results in a decrease in diameter and increase the blood pressure. So that will help fix part of the problem, but this is not enough. Okay, the vasoconstriction alone is not going to be a permanent solution. It will start making you feel better, but you really need something more long-term. So the second action of angiotensin is to go to your adrenal gland. Okay, it's going to go to your adrenal gland. Remember your kidney, right? Your kidney is wearing a party hat, and the party hat is the adrenal gland, right? Okay. So it will cause the adrenal gland to secrete aldosterone. Aldo like the brand, and then sterone. Okay, so it ends with ONE, like testosterone, progesterone. It is a steroid hormone. So what does aldosterone do? How does it help increase our blood pressure? Well, aldosterone is going to go to your distal tubule and collecting duct. Remember earlier in the table, we said the distal tubule and collecting duct, they are, for the most part, impermeable to substances. And when things do get through them, it has to be controlled by hormone. So aldosterone is a hormone. So what will it do at the distal tubule? Well, it will increase the sodium and chlorine absorption. Okay, remember, absorption means going in what direction? Blood to filtrate or filtrate to blood? Think about that for a second. Absorption. Filtrate to blood or blood to filtrate? Anybody? Filtrate to blood or blood to filtrate? Okay. Absorption is to take it back to the body, right? So Tina, you're correct. We were taking it from the filtrate back to the blood. Okay, so here is your filtrate. Okay, you don't really have to draw this. You can just watch. This is the blood. Okay, and what are we taking back? We're taking sodium chloride, right? So we'll take these sodium chloride and then we'll move it back to the blood. So now the blood has more sodium and chloride, right? If you increase the osmolarity of the blood, then the next thing that will happen is water will also be absorbed. By osmosis. Okay, the water always follows the salts, right? So if aldosterone increase the amount of salt that's going back to the blood, water will also be sucked back into the blood. And if you have more water in the blood, you will increase blood volume, and then you will increase blood pressure. Okay. So that's the RAS system. Right? After going through all that stuff, it's gonna bring your blood pressure back up. Now, if you have a phone or something, if you if you have copied this down, fantastic. Keep it in front of you. If you haven't, maybe just take a picture of this part right here. This part right here, the part that I'm highlighting right here. Just take a picture of that and keep it handy with you. I'm gonna ask you a question right now, but you need to have this part here, okay? I'm gonna give you a moment to do that, and then I'm gonna throw you the question. The question will be on the quiz, so make sure you understand it. We ready? All right, I gotta find a question. This one. I'm gonna launch the poll. Okay, please try it. It's okay if you don't get it right the first time. I'll tell you how to do it. Okay, I'm just gonna give you another 15 seconds or so to think about it. And then we'll take it up. Okay, well, thanks everyone for participating. We have answers all over the place, and D seems to be the popular one. So let's take a look at how to do this question. Okay, so to do the question, we're gonna have to go back to this thing that we just drew, and hopefully it will make sense when I try to explain it to you. Oh my goodness, what's happening? There we go. Okay, so I'm just gonna try to put it here and see. So the question says, what does it say? The level of renin is high, high renin, but ACE is low. Okay, so we have a lot of renin, but we don't have we have low ACE. So I'm just gonna cross it out. Okay, low ACE, cross it out. So let's see what happens. You start with angiotensinogen. If you have high renin, then angiotensinogen will be converted into angiotensin number 1, right? Okay, so this reaction will keep on going until you run out of angiotensinogen. So if you have high renin, this is going to be low because you're using all of it and you're converting it into angiotensin number 1. And to go from 1 to 2, we would need to have ACE. But the question says we are low in ACE, not enough ACE. So without ACE, the angiotensin number 1 will be stuck here. We will not be able to go forward. So this will be high because it's not going anywhere. And we're not forming any angiotensin number 2, or we're forming very little of it. And so this is going to be low. So the correct answer is low angiotensinogen, high number 1, and low number 2. So D is the correct answer. Does that make sense? Okay, please ask if you want me to explain again. All right, let's try another one, huh? Okay. What if I say we have low renin, and high ACE? Low renin and high ACE. Okay, low renin and high ACE. I want you to think about what the answer would be. Okay, sorry, it's a mess here. Hopefully you can see. All right, I'm gonna relaunch the poll. Can I what? Oh, repeat? Yeah, okay, sure. But let's try this out first. Okay, see if we can get this right. Okay, so I'm changing the question. I'm saying, what if we have low renin and high ACE? Then what's going to be the answer here? If the answer is not here, then choose E or something. Okay, choose E if it's not here. All right, let's take this up. Okay. Remember, a question like this will guarantee to be on your quiz. Guarantee. So make sure you understand it. All right. So here I'm saying low renin and high ACE. Right, so let's do this a step at a time. We start with angiotensinogen. With low renin, am I gonna be able to convert it into angiotensin 1? Is this reaction gonna happen if I don't have a lot of renin? Yeah, I mean, it's going to happen a little bit, right? But like, you're not going to do this. You're not going to be able to do this like at a very high level. So because of that reason, you will have a lot of angiotensinogen. You're not using it up, okay? Because you don't have enough renin, okay? And so you will have a little bit of angiotensin 1, but it will relatively be low, okay? It doesn't matter. You have a lot of ACE. Okay, if you have a little bit of angiotensin 1 and all of it get converted into angiotensin 2, it will still be low, right? This is not going to be high just because the reaction is going on, because you don't have enough raw material to begin with, right? So, the bottleneck, right, is the renin. That's why we would have a lot of angiotensinogen, low angiotensin 1 and low angiotensin 2. The answer is B in this scenario. Okay, so you have to see where the bottleneck is. Basically everything before the point that is low is going to build up. Okay, and after that, nothing really happens. It's going to remain low. It doesn't matter if you have something, if the ACE is high over here, because again, where we don't have the raw material. Do you understand? Yeah, Hima, are you okay now, or do you have a follow-up question? Oh, you okay now? Okay, all right, good. Yeah, just think about this. And if you're still confused, feel free to ask me later on. Okay. The kidney was gone. All right. So this is not the end of it. Okay. Now, in the body, there are usually some like balancing forces, if you would. Okay, some yin and yang going on. So if we have the ROS system to increase the blood pressure, you probably will be thinking like, oh, what happens if the blood pressure is too high? Is there something that could bring it back down? And you would be absolutely correct. Okay, so I'm gonna do this in hot pink over here. If you have an increase in blood pressure, and that could be caused by various factors, okay? Like you might have too much fluid in your body. You might be stressed out chronically. You might have some kind of cardiovascular problem, right? A bunch of reasons can lead to your you having high blood pressure. And so in the heart, in the atrium of the heart, there is a special group of cells that will be able to detect the high blood pressure. Okay, and when they detect the high blood pressure, the cells will secrete something called ANH, the atrial natriuretic hormone. Okay. Atrial means the atrium of the heart. Natri is like natrium, which is sodium. Uretic means to pee it out. So this is a hormone that is secreted from the atrium of the heart, and it's gonna make you pee a lot of sodium. Okay, that's basically what it means. Okay, ANH. So when the body secretes ANH, couple of things happen. ANH will inhibit renin. So by inhibiting renin, based on what we just talked about, you would block the conversion of angiotensinogen. So angiotensin 1 goes down, angiotensin 1 goes down, 2 goes down, and basically none of these things are going to happen, right? Okay, and that's a good thing if you are experiencing high blood pressure. You don't want these things to be happening when you already have high blood pressure. Turning on the RAS when you have high blood pressure, it's just going to make it worse, right? Secondly, it is going to block aldosterone. Now, this might be a little bit puzzling for some of you. You might think, well, if I turn off renin, why do I need to turn off aldosterone? Because turning off renin should shut off everything afterwards, right? There are things that we haven't learned yet, but aldosterone can be released through another pathway. RAS is not the only pathway that causes aldosterone to be released. Later on, we'll learn that aldosterone can come out through other means. So just in case you have some aldosterone in the blood system, the ANH will shut that down as well, because we don't want any of these things to happen. And finally, the ANH will also go to the collecting, the distal tubule and the collecting duct. And basically, it will do the opposite of this. So now we have high blood pressure. I don't want the sodium to be in my blood. So instead of increasing sodium and chlorine absorption, we will increase sodium and chlorine secretion. Okay. And as a result, water will be secreted as well. Okay. And then you will decrease blood volume. You'll pee it all out, which would decrease your blood pressure. Okay. So ANH, ANH and aldosterone is an example of what we would call antagonistic hormone. Right? They have opposite effects, and it's the balance between the two that cause us to have normal blood pressure. Okay. So that's what's supposed to happen in the body. Right? If they work perfectly and when they balance each other out, we would have no blood pressure problem. But of course, we know for many people, blood pressure will be high despite the fact that we have ANH in the body. And when the internal mechanisms is inadequate in balancing the blood pressure, then we have to take medications. Okay, so for people suffering from high blood pressure, there are two types of medication they can take. We talked about the diuretic earlier in the semester, right? That pill causes you to pee, and by peeing more, you reduce your blood volume and lower your blood pressure. Another type of drug that people with high blood pressure or hypertension take is called an ACE inhibitor. If you know anybody who takes blood pressure pills, you can take a look at them. Okay, some of them actually will literally say ACE inhibitor on the box, and now you know what it is. The ACE inhibitor, when you take it, will inhibit the ACE. Okay, so by blocking ACE, you essentially stop the conversion of angiotensin 1 to number 2. Okay, so think about it, right? Like if you experience like chronic high blood pressure, your ANH will help you block renin, so low renin, okay? But, you know, even at low level, maybe some is still going to get converted into number 1. So you have the ACE inhibitor that would further block the next reaction, right? So you would actually have no angiotensin number 2, right? So in combination, the ACE inhibitor plus the ANH will collectively suppress the RAS system and to bring your blood pressure to control. Is that okay? Are we like any? I know it's really complicated, so I feel a little bit uneasy when nobody has any question. I guess it's just something that you have to like sit through it and think about it on your own and study a little bit. Yeah, but anything at all before we go on? Okay. Now I do want to write something else here to help you see the relationship of what's going on, right? So in the center here we have normal blood pressure. All right. So help me out here, please. If you increase blood pressure for whatever reason, right? If you increase blood pressure, right? What hormone's gonna come out to bring it back down? High blood pressure. We need to bring it back down. What hormone do we need? Who'd you gonna call? ANH, that's correct. Right, so ANH will bring the blood pressure back down by suppressing renin, suppressing aldosterone, causing you to pee more, right? It'll bring the blood pressure back down. If the blood pressure continues, if the blood pressure drops, dehydration, blood loss, what have you, we will activate the RAS, right? We'll activate the RAS, but ultimately, what hormone do we need to bring the blood pressure back up? What is the antagonist of ANH? Okay, renin is an enzyme. It's not the hormone, but that's the beginning. Yeah, the renin will then allow us to have angiotensin, which ultimately leads to the release of which hormone? That's right, aldosterone. I don't think there is a short form for aldosterone, though, so you kind of have to write it all out. Does this make sense? Like the two balances each other out. And I need you to understand this theme because there are other stuff in the body that does this, and we'll learn about it, right? Like insulin and glucagon, you might have heard of them. They maintain normal blood sugar level, okay? So this will be applied to future lesson as well. All right, so this aldosterone is activated by the RAS system, right? Okay. All right. I think that's where we're gonna stop. If I go on any further, I think it's it's just gonna be very exhausting for you guys. These tables are very important, and I filled them out already in the lecture recording. All right, so just fast forward to that part and fill it in yourself. Someone was telling me that they watch my lecture recording at 2 times speed. Which actually is not a bad idea, okay, if it's kind of slow for you. But if you're skipping ahead, when you get to the hormone chart, make sure you slow down and fill it in, okay? Because it tells you where it is secreted from and what triggers it and the effect. For example, just try one for an example. Aldosterone. Where is aldosterone coming from? What organ? What structure does the aldosterone come from? Yes, lecture recordings are like for the whole course is posted already. Okay, Ahmed. That's right, adrenal gland. And Mavis did it, one step further. The adrenal cortex, exactly, the outside of the adrenal gland, right? Very good. And ADH, which is something we haven't talked about yet, but ADH is anti-diuretic hormone. Diuretic means pee, right? So this is like anti-pee hormone. It makes you not pee. When do you want to pee less? Think about it. When do you want to pee less? What's a good time to not pee? It's a strange question, right? When I phrase it that way. Yeah. At night, that's right. So when you're sleeping, you don't want to wake up and pee. So your ADH level is going to go up. But, are there any other scenario that you find yourself in where you don't necessarily want to pee? You want to conserve water, in other words. Yeah, in a desert, right? When you're dehydrated, exactly, Victoria, right? So when you're dehydrated, when you're not drinking water, your body actually produces ADH. And that helps you conserve the water. Okay, so make sure you read that as well, watch that part of the lecture as well. And I'll post the fill in the blanks answer for you as well. Okay, so like I said, for those of you who missed it, the quiz is next Tuesday. I will give you 2 attempts. Okay, and the best one is the one that will be recorded. All right. Before you go though, let's do a few more poll questions. Because these are kind of important ones. Okay, so please try this one. Kidney diseases and buffers. Could be, but for kidney diseases, it might be like you know what? I'm just gonna do you guys a favor. Okay, no kidney diseases on the quiz. Okay, no kidney diseases. Just gonna take that off. All right, so one less thing for you to worry about. All right, how are we doing here? I only have 6 people who've answered this question. This should be a very straightforward question. Which of these things, these activities, will cause you to activate a RAS? When you drink several glasses of water, will that activate a RAS? Shuffling the driveway for several hours on a cold winter day, will that activate a RAS? What do you need to do to activate a RAS? Okay, that's right. When you're mowing the lawn for several hours on a hot summer day, that will definitely activate the RAS. And the reason? Well, because on a hot summer day, it's going to make you sweat a lot, right? And if you don't drink enough water to replenish, you will be slightly dehydrated, your blood pressure might drop, and we need to activate the RAS. And you're like, but what about shuffling the driveway for several hours? You would sweat too, but you sweat less on a cold day, right? Okay, so the key word here is hot summer day. Understand? We did this one. This one is gonna be on. Okay, I'm gonna tell you again. Let's do a multi-select. Okay, some of you are a little concerned about the multi-select all the time. Oh, sorry, I launched the wrong poll. Multi-select. Let's try again. Okay? So to answer this question, this is the picture you have to think about. That's the renal pyramid, and we have the nephron right here. And if we're damaging the medulla, everything that's in the medulla will be affected. So Bowman's capsule is okay because it's in the cortex. Proximal tubule is in the cortex. The loop of Henle, right, is going to be affected. The distal tubule is okay. Collecting duct is going to be affected. Okay, so only two things, right? Loop of Henle and collecting duct. Because all these things, as you can see in the picture, Bowman's capsule, proximal tubule, distal tubule, glomerulus, they are all in the cortex, right? They're not in the medulla. So they will not be affected. Just the first one and the last one. Okay. Yeah, this one is very long. Let's take a look as well. Go ahead. This is multi-select. And again, ADH, I only explained briefly, but this conserves water. Okay. You know, maybe I'll just take this one up for you because I didn't explain much about ADH. The thing you need to know is for ADH, it acts on the distal tubule and the collecting duct, and you absorb the water in those parts. So let's take a look. ADH monitors appropriate osmolarity by reabsorption of water, and RAS maintains osmolarity by stimulating Na plus reabsorption. So both of these are actually correct, okay? You would reabsorb the water to conserve water, so that's correct. RAS will reabsorb Na plus, that is also correct, right? By reabsorbing the Na plus, you will increase blood volume and increase blood pressure, so that's correct. Next one, ADH and RAS work antagonistically. ADH stimulates water reabsorption during dehydration, and RAS removal of water when it is excess in the body. That's not correct, okay? The RAS does not remove water, right? RAS conserves water. Both RAS and ADH help conserve water to bring the blood pressure up. So this is not correct. And ADH is not antagonistic to RAS, right? ANH is the one from the heart. ADH will be released and RAS will be activated when a person is severely dehydrated. That is correct. Severely dehydrated, you need to conserve water through ADH, and the RAS will help you maintain blood pressure. Only when they're together at the proximal tubule will reabsorption of essential nutrients back to the blood takes place. That's not true. Okay, reabsorption at the proximal tubule happens regardless. Okay, the RAS and the ADH, they act on the distal tubule and the collecting duct way down at the end of the nephron. Both stimulate the adrenal gland to secrete aldosterone. Not true at all. ADH has nothing to do with aldosterone. Only the RAS will result in the release of aldosterone. Okay. One last part here. Diuretics are drugs sometimes used to treat individual with high blood pressure or hypertension. These drugs inhibit the reabsorption of Na plus by the loop of Henle. What effect would this have on the osmolarity in the nephron? Okay, so over here, remember we drew this diagram. Okay, so on the way down, we are going to be absorbing water. Right. So earlier I showed you, right, the urine color, the filtrate color will get very concentrated like that. Okay. Now on the way up, you were supposed to absorb NaCl And as you absorb NaCl, it should get lighter again, right? But the drug here blocks the reabsorption of NaCl, right? So that means it will stay concentrated because you're not taking back the NaCl. So let's see which one of these answers match that explanation. Osmolarity would be higher than normal in the collecting duct. That's the correct one, okay? Because now we are blocking all the absorption of NaCl. We will be causing the concentration to not get diluted as it goes up, and so that would be the answer. Okay, osmolarity would be higher than normal in descending limb. This has nothing to do with the descending limb. In the descending limb, it is only water that's being absorbed. So if you're blocking the reabsorption of Na+, it will not affect the descending limb. Osmolarity would be lower than normal in the proximal tubule. Again, it doesn't matter because proximal tubule is all the way over here before any of these, before the loop of Henle, right? So if the drug targets the loop of Henle, it will have no effect in the proximal tubule. Osmolarity is lower than normal in collecting duct. Well, this is the opposite of the first one. So if the first one is correct, this one is not correct. All right. Okay, so a very heavy lecture, I understand. Please take some time to study, and all the best for Tuesday's quiz. Now, next week is a little bit complicated, okay? Oh, how about application question? I don't know if you noticed, but these are application question, right? Like this drug thing here, that's application question. These are multi-select, and this would be application question, right? Because you have to think a lot, right? It's not just things that we can look up in the notes, right? I hope that helps a little bit. Next week is a special schedule. Okay, we have to meet 2 times. Okay, we have to meet on Tuesday and we have to meet on Thursday. And the reason for that is because the lecture 10 has muscle part to it and then the bone part. The lecture recordings only have the bones. It doesn't have the muscle. So I have to teach you the muscle when we meet up. Okay, and also I think we are a little bit behind, if I am correct. Yep, because we were supposed to do this lesson, the kidney, last week when we do the test review, okay? But I decided to postpone it so you can focus on studying. So now we have to catch up. So next Tuesday, we will do the muscle and the bone together, that lesson. And then on the Thursday, we will do the reproductive system. I don't know if you know, but we only have 2 or 3 weeks left before the course is over. Okay, so we're in the final stretch. So just hang in there. Okay, make sure you come for both Tuesday and Thursday next week. All right, thanks again, and I hope you had a good week. Bye for now, guys.